let's see here, the heredity means the transfer of biological character from one generation to the next. It is called as heredity. In another way, we can say that the transfer of biological character from parents to offspring, it is also known as heredity. So you know about that. Who is the pioneer of modern genetics? Tell.
John George Mendel was the pioneer of modern genetics.
Yes, she is correct. John Gregor Mendel is the pioneer of modern genetics, and around the period of 1886, it took a long time for him to understand the research of conclusion about his research about heredity. And in the period of 1902, the reason behind the sudden changes were understood due to the mutational theory of Hugo. And in the period of 1901, the scientists Walter and Sutton observed the cell of pair of chromosome in cells of grasshopper until it was not known to anyone. And that time they started research in the direction of to finding the nature of genetic materials, and they proved that every genes are carried via chromosomes. And in the period of 1944, the trio of scientists, Ausfall Avery, Maclean McCarthy, Colin McLeod, they proved that except viruses, all living organisms have DNA as a genetic materials. And in the period of 1961, the French geneticist Francois Jacques Monod, they proposed the model for protein synthesis with the help of DNA in the cells of bacteria, and it helped to remove the uncover the genetic codes that hidden in DNA. Now we learn about in what way the hereditary science is useful for us. Anyone tell me in what way the hereditary science is useful for us? Yes?
For to cover various genetic disorders.
Yes, yes, you are right. The hereditary science is useful for us diagnosis, treatment, and the prevention of hereditary disorders. And it is also useful for reproduction of hybrid varieties of plants and animals. Now we learn about three topics. The first one is transcription. The second one is translation. And third one is translocation. You know that firstly we learn about transcription. What is transcription? You know that the process of protein synthesis by DNA through the RNA. This is called central dogma. Besides, there is a uracil in present in the RNA instead of thymine of DNA. This process of RNA synthesis, it is called transcription. Now our transcription process is completed. Then we move towards the process of translocation. Firstly, we need to understand what is translocation. The mRNA is made up of thousands of triplets codon, and these triplets mRNA is made up of thousands of triplet codons, and according to the message of mRNA, the amino acids carried by the tRNA. For this purpose, the tRNA having the anticodon for the complementary sequence on mRNA. This process is called translation. Now translation is completed, then move towards the translocation. In previous standard, we have already learned about translocation. Translocation means the process of conducting food materials, that is called translocation. Now our translocation process is also completed. Then we move towards the evolution. Firstly, we need to understand what is evolution. Evolution is the gradual changes occurring in living organism over a long duration. Let's write here the definition of evolution. So now we learn about evidences of evolution. There are how many evidences of evolution explanation given in our textbooks?
Yes.
Six. There are six evidence of evolution. Evidence of evolution says. Now we write them one by one. The first is morphological evidence. Then second one is anatomical evidence. Then third one is, can you tell me? Yes, correct. And the fourth is, tell me. Yes, correct. Then fifth is and then this last one is firstly we learn about morphological evidence. What is morphological evidence? In morphological evidence, there are various similarities between animals. Their structure of nose, structure of mouth, structure of eyes, their structure of face, etc. are included in morphological evidence. So our morphological evidence is completed. Then we move towards the anatomical evidence. In anatomical evidence, there is many similarities between the structure of bones of the structure of bones of animals. That is called anatomical evidence. Then we will learn about vestigial organ. Firstly, we need to understand what is vestigial organ. The degenerated and useless organ of our human body, that is called vestigial organ. So, can you tell me the name of vestigial organ one by one? Yes, appendix. Then next,
Wisdom.
okay. The first is appendix, then pinna, then wisdom teeth, then tailbone. You know what? The appendix is useless organ of our human body, but it is also useful for ruminant animals. And then we learn about and pinna. Pinna is also useless organ for our human body, but pinna is also useful organ of monkeys. And then next our tailbone and our wisdom teeth is also useless organ of our human body. So all evidence is completed. Then we move towards the next topic. The next topic is DNA and RNA. Previous standard we have also learned about that, but now let's revise that topic 3 again. What is the full form of DNA? Tell me.
It's ribonucleic acid.
Deoxyribonucleic acid. Firstly, we learn about DNA. The chromosome is mainly made up of DNA, and this acid was discovered by the Swiss biochemist Friedrich Mister in the period of 1869 while the studying of white blood cell. And you know what? The DNA is represented in the nucleus of the cell, hence it was name given to the nucleic acid. And you know that the all living organisms have a DNA molecule like viruses, bacteria, and humans. They control the functioning and growth and division of the cells. That's they are called master molecule of human body. And then we learn about the model of DNA. The Watson and the scientist Watson and Crick. The Watson and Crick produced the model of DNA as per this in the period of 1953. As per this model, the two parallel threads of nucleotides coil each other. This arrangement is called double helix, and we can compare this arrangement with the ladder and coil. Then we learn about RNA. What is the full form of RNA? Now we learn about RNA. RNA is the second important nucleic acid in our human body, and it is mainly made up of ribose sugar and a phosphate molecule, and it is also made up of four nitrogenous base. First is adenine, then second is guanine, third is cytosine, and fourth is uracil. DNA is also made up of four nitrogenous bases: adenine, guanine, cytosine, and last one is thymine. In place of thymine in RNA, there present is uracil. So let's move. So there are three types of RNA. Can you tell the name?
Transfer.
Firstly, we have take rRNA. Then mRNA. Last one is let's draw the diagram of rRNA. Firstly, my diagram is not perfect. Let me understand. This is the diagram of rRNA. Then we draw the diagram of mRNA. This is the diagram of mRNA. Now let's draw the diagram of tRNA. So here the diagram of types of RNA is completed. Then move towards the next topic, chromosome. The structure of nucleus in the cell that carries the hereditary characters, that is called chromosome. Chromosome is mainly made up of DNA and protein, and chromosome shaped like a dumbbell shape. And there are how many types of chromosome?
4.
Tell me. There are 4 types of chromosome. The first is metacentric. Then second is submetacentric. Then third is acrocentric. Then fourth one is telocentric. Now we learn about the chromosome types of metacentric. Metacentric means the centromere is at the midpoint of this chromosome. Therefore, chromosome look like an English letter V. Which letter? Tell me which letter?
V.
Then we learn about submetacentric. Submetacentric. The centromere is at the near of this midpoint of this chromosome. Therefore, chromosome look like an English letter L. That's called submetacentric chromosome. Then we learn about acrocentric chromosome. The centromere is at the near of this chromosome. Therefore, chromosome look like an English letter J. Then we learn about telocentric chromosome. Telocentric chromosome means centromere is at the right end of this chromosome. Therefore, chromosome look like an English letter. Which letter? Let's draw the diagram of metacentric chromosome. This is the diagram of metacentric chromosome like a V shape. Then let's draw the diagram of submetacentric chromosome. This is the diagram of submetacentric chromosome. Let's draw the diagram of acrocentric chromosome. Then let's draw the diagram of telocentric chromosome. This is the diagram of telocentric chromosome. This is the diagram of acrocentric chromosome. Then we learn about to find the number of chromosomes in each organism. Each organism. First we write here serial number 1, 2,